Thanks everybody for coming out for this early talk. I hear there's coffee out there. It's pretty good. As Susan said, my name's Matt. I'm going to be sharing updates from the Great Lakes Mass Marking Program on salmon and trout. I'm going to start with an overview of our program and then highlight some of our major findings to date for Chinook salmon, lake trout, and for steelhead. So getting right into it, our program is also known as the Great Lakes Fish Tagging and Recovery Lab. We are a collaboration among federal, state, and tribal agencies that's coordinated by the U.S. Fish and Wildlife Service out of the Green Bay office. There we go. We got some advancement going. We began in 2010 to address questions on the survival and natural production of salmon, as well as other measures like growth and movement. We have been funded since that time through the Great Lakes Restoration Initiative, which is non-permanent funding that's funneled through us via the EPA. And over this time, we have collected 13 years of data on over 178,000 fish from open water angling. Our program has 4 major divisions. We have the tagging and marking of hatchery fish. We'll get into more detail on all of these shortly. We also have data collection and tag recovery from the sport fishery. We also do some work when it's not field season in the lab. We're extracting tags from these heads, aging wild fish, and processing some of the other samples that we collect. And then, of course, we have data analysis and support for fisheries management. So taking a closer look at our history of tagging, this is a figure showing the number of fish marked in millions by year since the inception of our program. Our latest year here, 2025, you can see we had 3 major species, tagging a little over 3 million lake trout, a little over 4 million Chinook, and a little over 2 million coho salmon. But over the course of this study, we started with lake trout. We've been doing lake trout throughout. Chinook salmon received tags through here. We continued to clip them to identify wild and hatchery fish, but those resources for tags went to steelhead, which is what we see in pink. 2024 is the last year of steelhead tagging, but we are still collecting lots of data on those fish in the fishery. So one thing I'm commonly asked is to learn a little bit more about the tagging and marking process. I'm going to actually have to click on that video. I don't know if somebody with the computer can actually click on that. Thank you. So this is inside one of our tagging trailers. We have some fish that are in a big bin down there. We bring them in from the raceways. They're in here. This is called a length sorter. They're going to be rapidly measured in length to within a fraction of a millimeter by this optical system. You can see these red lights that's actually taking very rapid measures and then dividing these fish into 6 different tagging lines that are calibrated for tight size ranges. You can see those lines there. My kids love this video because the fish get to go down the water slide. At least that's what they said when they were 3. But you'll see they're in now separate holding pens based on the size. They are swimming into this volitionally because of current going upstream. And then this is the actual clipping apparatus. If you look really carefully in the middle there, you'll actually see a fish appear right there. And that little brown thing is coming and clipping the fin. At the same time, the blue boxes at the front are what's injecting the tag. And then those fish go through a quality control device. Basically, there's a before and after picture verifying a fin clip and a magnet verifying tag. If both of those things are successful, it gets diverted back right out to the hatchery with those fish never leaving the water. So that is the tagging process. These tags have a 6-digit batch code, corresponds to where a fish was stocked, its genetic strain, its year class, hatchery of rearing, and that allows us to answer a whole bunch of questions that are of interest to management, but only if we have data on those fish as they return as adults. And so that's the second part of this program, where we have a large-scale collection network on Lake Michigan in the U.S. waters of Lake Huron. These black stars are where we have historically stationed our teams of biotechnicians, and then the red dots are where we typically sample. We are actually adding a team in Muskegon here this coming summer at the Annis Water Resources Institute, so thank you very much to them. In 2025, we actually had a little bit of a different sampling network. We did have somewhat reduced staffing levels and had teams stationed at these locations, but we did have lots of travel into Michigan to try to keep a spatially robust design in 2025. 2026, it looks like we're going to have a better coverage closer to what we had been doing historically. The third major component of our program is back in the lab. This is not automated. We have to pull these tags out by hand. We have close to 160,000 recovered thus far. Here's a blown up image of a tag. If you have really good eyesight, you can read the tag number. My eyesight's not that good, but trust me, it's there. And in addition to this, we also want to get age data on wild fish. That provides a lot of good measurements for our catch at age models, as well as for looking at the proportional length at age for assessing wild recruitment. And then lastly, I just want to kind of summarize the stuff we do for fisheries management. A lot of our data are tied right into management models, like the PPR ratio and things that are used to make policy and stocking decisions. But on top of that, because we have this wide existing collection network, we've also been able to partner on a lot of cool studies led by others, including looking at sea lamprey wounding rates on various species. We've been a part of several of the studies on salmon and trout diet, including collecting in last year's data. We've partnered with researchers at Central Michigan to look at several different species of Pacific salmon and the natal origin of those wild fish, something we did with coho salmon last year. Collecting samples to look at the genetic composition of things like wild lake trout. We started to do that for pink salmon with Joe Langan and some of his colleagues. Biocontamination by PFAS, and then some new methodologies such as looking at DNA from steelhead to try to determine if that can be used for assessing age composition. All these are partner-led, but again, we're putting our hands on so many fish, it allows us to kind of lend a hand with these things. So I just want to highlight that before I go into our results. So I'm going to start with Chinook salmon. One of the things we do every year is try to estimate the wild production in Lake Michigan. This is done by looking at the observed percentage of wild fish recorded at age 2 in a given year, and we use that observed percentage with the known number of stocked fish to figure out how many wild fish must have been there to produce that percentage. I will say that we take data in the hatcheries at the time of tagging every 2 hours. Those data suggest 99.5% of the fish are leaving the hatchery with a fin clip. We do account for that 0.5% error in these numbers, but it is a pretty good indication if you're out on the lake and it has an adipose fin intact that it is going to be a fish of wild origin, if it's a Chinook. This figure is showing our time series of Chinook salmon wild production. Smolts in millions versus the year class. Our orange bars are the known number of stocked fish, and the blue bars are the estimated number of wild fish. So we can see, just to highlight the most recent numbers, at age 2 for the 2023 year class, we're at a little over 11.5 million. With 8 million wild fish coming in. We do an estimate at age 1 that'll be updated next year for 2024 with 2026 collections. We can see that our projected estimate's down a little bit. But one thing I want to highlight, if we look at the whole time series, is the differences in year class strength. And to emphasize that, I'm going to take those stocked fish off of this figure. So we're left with just the wild fish. And if you kind of look at this the way I do, I see kind of 3 main periods of patterns here, even though there is, annual variability. We do have these first 7 year classes where wild production ranged from 6 to 8 million fish. We then had a very weak year class in 2013 that was caused in part by substantial drought conditions during the fall of that spawning year, followed by a protracted and long winter. And following that year, my best hypothesis is, because of that weak year class, we had fewer wild spawners returning, and as a result, we had a period of time with only not a handful, but 3 to 5 million wild fish. That's way more than a handful, but it's less than 6 to 8 million. But we have seen with the most recent year classes an increase back to the kind of pre-2013 year class levels, where we're back in that 6 to 8 million per year range. Other things we've learned from Chinook salmon, I know I've been talking about survival for several years now. These findings are published now in the North American Journal of Fisheries Management. The key finding here is, if you look at this figure, this is a heat map of Chinook salmon survival. The warm red colors are higher survival. These kind of pale yellow colors are lower survival. So we've observed higher survival here for fish stocked in the Wisconsin shoreline, but really low survival for fish stocked in Green Bay, in part because of predation and high water temperature, and in MM6, we think because of predation in the river there prior to those fish exiting from their stocking location at the egg take facility for Michigan. I can tell you, I don't have the figure, but we did see a decline in relative survival of stocked fish as well from 2011 to 2016. And now that we're tagging Chinook salmon again, starting this year, we should be able to reevaluate and see where things stand close to 10 years later. One thing I want to highlight about these results is that this information can help assess stocking programs and maximize return on taxpayer and fishing license dollars. I do know that Wisconsin is making some adjustments to their stocking plan, in part driven by these results. Another thing we've learned about Chinook salmon is that they do exhibit natal homing. This is a composite figure of all of our recoveries in the state of Wisconsin. I want to call your attention to this red line. These are fish that were stocked in the same unit where they were caught. That's less than 10% throughout most of the year. So we do say that Chinook salmon are pretty much being caught by everyone, everywhere for most of the year, but we can see that they are returning to near where they were stocked by the time we get to the spawning season. Now, I did mention this is for Wisconsin. We see about 2/3 wild. That's declining in Wisconsin as we get into the spawning period. I don't think I have the figure, but in Michigan, this is about here and then goes up to about 100% as we get into the spawning season. So that motivated us to look at the collections of wild fish, our wild relative abundance rates for Chinook salmon in the fall, and that produced this heat map. Basically, we see a higher catch rate of wild Chinook salmon in the fall in northeastern Lake Michigan, and given what we know about natal homing, that suggests that that's a focal point of wild production. So a lot of these results are also recently published in a North American journal if you want to learn more. Second, I want to talk about some of our results with lake trout. One of the things we do every year is look at the relative abundance of wild fish to kind of track trend lines in that. And so this is our measure of relative abundance. We look at the number of fish observed by our sampling teams per day in the field, and we're interpreting that as a relative abundance pattern. For lake trout, I boiled the results down into 3 large regions today. We have southern Lake Michigan, which is this dark blue unit with 8 districts, northern Lake Michigan, which is this light blue unit, and then Lake Huron, which is orange. So without further ado, this is a figure showing our trend lines in wild lake trout relative abundance in Lake Huron here in orange, southern Lake Michigan in dark blue, and northern Lake Michigan in light blue. And the main takeaway from this figure is in all 3 of these regions we have seen increases in the relative abundance of wild lake trout in the sport fishery over the timeline. But I want to kind of dive into these results in a little bit more detail. First, I know this isn't the Lake Huron session, but I'm going to look at Lake Huron real quick. In this case, we're looking at relative abundance. The wild fish line is going to be in gray for all of these figures. The hatchery lake trout will be in orange, and then our total, which is basically a sum of these two, is in black. So the take home here for Lake Huron is that for some time now we have had a greater relative catch of wild lake trout than hatchery lake trout, and you can see that that is driving the general trend line in our catch rates for lake trout in the Huron sport fishery. It's a bit of an opposite story in northern Lake Michigan. We have seen an improvement here from near zero relative abundance of wild fish to higher than near zero. So that is a success story, but even so, there is still a lot of hatchery lake trout in northern Lake Michigan, and our total catch rates there are really reflective of the hatchery trout rather than the wild trout. And then lastly, in southern Lake Michigan, we have this interesting pattern where we actually see an increase in wild lake trout with a corresponding decrease in hatchery lake trout. This decline in hatchery lake trout appears to be tied to a time lag of about 6 to 10 years to stocking cuts in southern Lake Michigan. So that is an expected decline. But you can see over the course of this time series, the southern Lake Michigan fishery has changed from largely being reflected by the hatchery lake trout to at this point kind of really incorporating both of the patterns here. So that's a long and short way of kind of describing what we're seeing in the sport fishery for lake trout. I do want to highlight another paper that came out this year. This was an observation made by biological technicians finding some lake trout that were in spawning condition in the spring. In total, 9 female and 2 male lake trout from April through July were found to be ready to spawn. And this was documented through histological analysis as well. I just want to mention this because these fish were both hatchery and wild origin, multiple strains, age 6 to 19, and I do know that this has been of interest to the Lake Michigan Lake Trout Working Group, in part because others have been observing this pattern as well, especially in the Southern Refuge and in the Traverse Bay area. So something to keep an eye on. We don't know, I mean, this is obviously still kind of a small, very small portion of the lake trout out there in all likelihood, but it may offer some adaptive advantages, including reduced competition with fall spawners or lower predation risk for juveniles. We've also recently updated a matrix of lake trout movement throughout Lake Michigan. This was an update of a 2019 analysis to really kind of look at older aged fish as well as increase our sample size. This is one of the products we produce that feeds directly into the modeling efforts for looking at the catch at age models for lake trout throughout the lake, basically looking at migration rates in and out of the various stock assessment units. We didn't have any major changes to the main conclusions since 2019. So what those conclusions were, we still see that about 80% of lake trout are being caught within 60 miles or 100 kilometers of where they were stocked. So they are not moving as much as salmon in the lake. Nearshore stocked lake trout are very rarely crossing to the other side of the lake, but offshore stocking locations in the refuges, as well as Julian's Reef in Illinois, do continue to support fisheries nearshore lake-wide. We also see that most fish are remaining in the assessment unit in which they are stocked. But one change that we did see from 2019 is for fish stocked on the Southern Refuge. This figure is the analysis in 2020. This is 2025. In both cases, you're looking at the percent recovery of fish stocked on the Southern Refuge, which is this yellow unit. So in 2020, we saw about half of those fish recovered in the Southern Refuge and about half distributed throughout both shorelines. In 2025, however, we saw close to a little over 80% up there in the refuge, only about 20% moving lake-wide. And the main reason for this is something that was alluded to yesterday in several of the talks, and that is the emergence of the Klondike great trout population there on the Southern Refuge. And I can demonstrate this pretty clearly with this comparison here. So these are both from the 2025 analysis. These are just our Klondikes. We had a little over 2,800 recovered. 99% of those were recovered in the Southern Refuge. So those Klondikes are really by and large staying put. All of these zeros do add to 1%. They're just less than 0.5, so rounded down. We contrast that with our Seneca our Seneca lake trout. Here we only have about 24% of those fish on the refuge. So those Senecas actually are moving. We see a very distinct pattern between these two strains, those Senecas contributing to fisheries on the near shore. And then lastly, I want to highlight some of our results from steelhead. I do see that yellow line, so we're going to be going through this a little bit quickly. But we have seen pretty consistent estimates of wild production for steelhead, ranging from about 32 to 50%. If you look at all these year classes, we're probably somewhere in the ballpark of 40% from coated wire tags, and that's pretty convenient because there was actually a completely different study using completely different methods that found about 40% wild as well. That's in an article by Ben Breaker et al. In Prep. So when we have two methods telling us the same thing, it gives us a good deal of confidence. We can use the same methodology we do for Chinook salmon to look at their wild production. Thus far, we've only been able to do this starting with the 2017 year class, and there really isn't much to talk about. The trend is pretty consistent with the exception that 2020 was a weird year for many reasons, and I don't think we need to extrapolate on that. One thing I want to highlight with the movement patterns of steelhead is that, like Chinook salmon, they are moving all over the place. This is an example map for steelhead stocked in Indiana. We can see them contributing through both of the shorelines. And so the main take home here is that everybody is catching everybody's steelhead. These are pie charts showing steelhead caught in Illinois, caught in Indiana, caught in Michigan, shores of Lake Michigan, and caught in Wisconsin with different stocking units represented by different colors. And while there are some differences, by and large, you're looking at the same pie chart and you're seeing that every state is relying on one another's fish for their fishery. We have looked at relative survival for steelhead as well. We found that yearlings do substantially better than fingerlings, with about a 2.5 times greater survival for yearlings compared to fingerlings. So when we look at the survival of steelhead, we're only looking at yearlings in this figure to kind of remove that effect. And in this case, we are seeing a higher survival of steelhead stock kind of in southeastern Lake Michigan, as well as the Wisconsin small tributaries. This is a tag lap that spans the entire coastline of Wisconsin with many different small rivers that are stocked. We do see low survival in Green Bay, like we did with Chinook, as well as in this particular unit. And then this is just a figure showing where those steelhead are stocked, so you can kind of compare stocking rates to survival rates. We have started to look at genetic strains as well. There are multiple strains stocked in Lake Michigan for steelhead. Thus far, the only trend we have seen is that for 3 districts where both Skamania steelhead and Michigan winter steelhead are stocked, we do see a statistically significant higher recovery rate for those Skamanias relative to the Michigan winter strain. So to kind of wrap that all up today, in conclusion, mass marking continues to be a Fish and Wildlife Service priority, and the tagging and recovery goals should be met in 2026. For Chinook salmon, we see somewhat stable wild recruitment over the past years, with higher survival rates in Wisconsin and a high degree of wild recruitment in northeastern Lake Michigan. We also see that those Chinook salmon are exhibiting lake-wide movement as well as natal homing in the fall. For lake trout, we do see that the catch of wild fish has kind of plateaued after this period of increase through 2021, but we are still seeing kind of positive news there compared to earlier on in the time series with very low evidence of wild fish. We had seen some evidence of spring spawning lake trout, as well as strong evidence that offshore stocking locations are supporting fisheries lake-wide. And then for steelhead, we are seeing still relatively stable wild recruitment, comprising about 45% of the fishery, with higher survival for fish stocked in Michigan, somewhat higher survival for Skamania strain, with lake-wide movements showing that all states are relying on one another's fish. With that, I'd like to thank you all for your attention at this early hour and use my last 11 seconds for questions.
anyone have any questions?
Just have a comment. Matt, really appreciate all the legwork you did to get a second biotech team in Michigan and in Muskegon. You know, I think Seth Herbst, I see him near there, he mentioned the Annis Water Institute